This video will present a research work on plant 3D phenotyping, focusing on how to follow a plant that grows over time in 3 dimensions, and how to measure this growth in 3D automatically using robotics and computer vision. To achieve these objectives that may sound simple, we have developed an important array of tools to complete an entire phenotyping workflow, from imaging a single plant in 3D to getting detailed results about its growth and development. This complex phenotyping workflow is performed thanks to a series of independent modules that can function both separately or in a fully integrated pipeline. The modularity of our components, which are 100% open source for both hardware and software, is a strong advantage for their diffusion and reuse in the plant phenotyping community in a potentially wide range of situations. So to begin with, we built a new robot, the ROMI Plant Imager. This robot is meant to image single-potted plants of mid-size thanks to a simple RGB camera that moves around the plant with a robotic arm. It's a simple cage with limited space footprint that does not require special environment or conditions to function, so it is adaptable to a wide range of user contexts. Its design is very simple and made of open source hardware components which can be assembled as a do-it-yourself kit. This simple and modular hardware design is a key asset for such a phenotyping robot. First, it limits the price. The robot costs around 3,000 to 4,000 euros, at least 10 times less than current comparable commercial solutions. Second, simple design also means simple use. There are few components to connect and control. Only 4 CNC motors for the XYZ panoramic movements, a RGB camera, and a lighting system. Third, a simple design favors adaptability and evolvability. For example, we can image different plants with different architectures, different height from a few centimeters up to 40 centimeters high, or different thinness of their axis. We can also develop different parts of the robot separately, as we did recently with the arm. We are testing a new version with several Pi cameras. The robots generate a series of RGB images from all around the plant. Then, we develop a series of algorithms and programs to reconstruct a plant in 3D. All these tools are bundled in a single package that we called Plant 3D Vision. This is a Swiss Army knife for plant analysis in 3D, including different tools to process data all the way up to meaningful biological data. Like the robot, the architecture of Plant 3D Vision is the result of the fruitful alliance of biologists and computer scientists, of developers and end users. Since it is based on modular components, different analysis pipelines can be created. But we also developed and tested pre-built pipelines to help end users getting their desired results efficiently. Non-computer scientists will appreciate effortless installation and run through distributable Docker images and the automatic control of the execution of several connected tasks with a pipeline management system. But developers will also appreciate the open software opportunities to contribute to the project via GitHub access. Finally, we also develop a web app to gather and inspect all the results of your analysis. This interface, called the Plant 3D Explorer, allows to visualize all 3D objects that have been generated by the analysis, to display them as independent layers, and to check visually how they fit with the original 2D images acquired by the Plant Imager. The available content for display automatically adapts to the type of analysis that was performed. This tool is used both by developers to evaluate the performance of the 3D analysis of their tools, and for biologists to get the results of their phenotyping experiment. Using all these tools, we have obtained different phenotyping data and results. First, we can generate high-precision 3D point clouds of different plant species. Point clouds are very important 3D objects, since they are the input data of many downstream analysis tools. Our tools could thus be reused by many 3D phenotyping projects up to this point. A common computer vision task downstream of 3D point cloud is to identify the different parts of the plant based on their botanical nature: stem, leaves, fruits, flowers, etc. This task is called segmentation, and our tools propose different solutions to achieve it, based on different algorithms. Some are based on geometric and algebraic analysis of point clouds, some of them are based on machine learning. For example, our deep learning-based segmentation tool is able to automatically detect many fruits of this very complex branch Arabidopsis. Our solutions are quite robust, allowing the users to use different objects in the scene, like pots or tutor, to hold the plants. In this example, the tutor does not interfere with the fruit segmentation of the plant. Downstream of segmentation, precise measurements can be performed on the plant. In this project, we developed a pipeline to analyze the phytotaxis of Arabidopsis thaliana, a major organism in plant science. Here, the phytotaxis is the distribution of fruits around the stem. To study it, it is necessary to measure the divergence angle between successive fruits on the horizontal plane and the distance between their insertion point on the stem, called internode length. On our Plant 3D Explorer web app, you can see that in this example, the segmentation is 100% correct on the stem, and the automated measures on both divergence angles and internode lengths, in blue, is very close to manual measurement that were performed as a control ground truth. But with ROMI, we do it faster, with a much lighter workload. And now we will show how we can use our tools to follow the growth of a plant over time in a quantitative manner in 3 dimensions. This work has been published at the International Conference on 3D Vision 2021. Even if the station is not a growing chamber, not currently equipped to grow plants continuously, it is possible to make time-lapse imaging. More than possible, this type of non-invasive imaging is actually ideal to follow plant growth without damages and with minimal perturbations to the plants. In this example, we image a plant twice a day for 7 days. We did it for 5 plants. We could follow growth of the plant from bolting, the stage at which the main stem grows out of the flat above-ground rosette, up to the apparition of the first flowers and fruits. Despite the lack of automation to acquire time points, this demonstrates the ability of our plant imager to generate quality datasets of plant growth in 3D. Although this would require some development, the simple evolvable design of our device could be adapted in several ways to specialize in time-lapse imaging. One possibility would be to equip the cabinet for continuous growth of plants, dispensing for moving the plant in and out. A second possibility would be to couple it with robotics that would automatically convey plants in and out at a regular time points for imaging. More precisely, the challenge is the following. By comparing point clouds at 2 time points, we would like to measure plant growth at different scales, for example, for the entire plant or just a leaf. Also, we would like to detect newly formed organs. The finer the resolution at which we can map one plant onto another, the more accurate will be the estimation of growth, be it length, area, volumes. So it's necessary to perform this mapping at the scale of the point in the point clouds. Today, some tools already exist to match 2 3D shapes together. They function pretty well with organisms like animals or even humans, but they do not work with plants. Why? Because plants have multiple branches and leaves with similar geometric features, and they continuously grow new ones. These properties limit the accuracy and performance of traditional algorithms, which mostly rely on local geometric features. To overcome this limit, a recent approach proposed to create a skeleton from each point cloud, then establish a correspondence between the skeletons, and finally retrieve the correspondence between the points using the skeleton as a global spatial positioning system. However, the proposed skeleton matching method has limited efficiency and accuracy on plants with complex structures such as Arabidopsis. Therefore, we proposed a new framework for the space-time registration of growing plants. Unlike the previous methods, we do not establish the point-to-point matching on the whole plant directly, but we consider the shape correspondence at 3 different scales: branch scale, segment scale, and point scale. The proposed methods start with the creation of a topological skeleton of the plant at each time step. Then, these branches are further divided into smaller segments that possess a simple geometric structure. These segments are matched between two time steps using a random forest classifiers, a matching learning technique, based on their topology and geometric features. Then, for each pair of segments matched, a pointwise registration is devised using a non-rigid registration method based on a local ICP for iterative closest point algorithm. Then, merging back scales from the smallest to the largest, it is possible to obtain a pointwise matching at the level of the whole plant, and then for the entire time series. We applied our method not only to Arabidopsis, but also to young tomato and maize plants. The resulting data can be then exploited to measure growth. For example, here in this plot, we computed the growth of the main stem in blue that we can compare to the real measurement in green. In this second plot, we computed the growth of the different lateral branches, each branch with a different color. We then compared our method with the state of the art. Our results show that our approach achieves better or similar results with a shorter running time. In conclusion, this video showcases the main tools that have been developed by partners of the RAMI project concerning precise plant phenotyping in 3D. It illustrates some results and application, especially here for monitoring plant growth over time. Our tools are quite original in the field of plant phenotyping, and we hope that they will bring new opportunities for users with different profiles and background to use 3D plant phenotyping, be it in the lab for researcher or plant scientists, and maybe one day in the field for farmers.